Welcome to Computational Genome Biology. You finally decide to clean out that old, abandoned cabin or sweep the dark corners of the garage. As you step inside, you see a thick layer of dust stirred up into the shafts of sunlight. You take a single deep breath. A few weeks later, you are fighting for your life in an intensive care unit, your lungs rapidly filling with fluid. One breath introduces a silent pathogen into the lungs. This is hantavirus. In the spring of 1993, a medical crisis emerged in the Four Corners region of the American Southwest. An unusually wet winter, driven by El Niño weather patterns, flooded the arid landscape with new life. This abundance of vegetation and food triggered an explosive population boom in the North American deer mouse. Soon after, physically fit young adults began arriving at local hospitals in severe respiratory distress. They were dying of acute respiratory failure as their lungs filled with fluid. Medical investigators were baffled. The disease had a fatality rate of over 35%, yet doctors and family members weren't catching it from the patients. There was no evidence of person-to-person spread. Investigation revealed an ancient pathogen perfectly adapted to its environment, lying dormant in the dust of the high desert. Hantavirus belongs to the Buneviridae family. It is an enveloped, negative-sense, single-stranded RNA virus. Its genetic material is encased in an outer lipid bubble, and inside, the RNA is split into 3 separate segments. These viruses have co-evolved alongside specific rodent species for thousands of years, with individual viral strains intricately tied to a single type of host. Because of this shared evolutionary history, the rodents act as lifelong asymptomatic carriers. They host the virus indefinitely without showing any signs of disease. This relationship exists globally. While the deer mouse carries the virus in North America, the striped field mouse and the common brown Norway rat maintain different strains across Europe and Asia. As these asymptomatic rodents move through their habitats, they shed live viral particles through their urine, droppings, and saliva. A viral adaptation meant to create a harmless existence inside a rodent becomes a chaotic, lethal weapon when it crosses the species barrier into a human. The crossover happens through aerosolization. When dried rodent waste is disturbed, it breaks apart, launching microscopic viral particles into the breathable air. Once inhaled, the virus enters host cells through a process called receptor-mediated endocytosis. It specifically targets and hijacks endothelial cells, the delicate lining of our blood vessels. The resulting disease depends on geography. Strains in the Americas, like Sin Nombre, cause Hantavirus Pulmonary Syndrome, or HPS, aggressively attacking the lungs. The South American Andes strain is a rare exception here, as it is capable of human-to-human transmission. In Europe and Asia, strains like Hantan and Sol cause hemorrhagic fever with renal syndrome, or HFRS. This variation primarily attacks the kidneys, leading to vascular leakage and sudden drops in blood pressure. Depending on the specific rodent vector, hantavirus executes 2 entirely different, yet equally devastating attacks on the human cardiovascular system. Currently, there are no FDA-approved cures, specific therapeutics, or widely available vaccines for hantavirus infections. Because early symptoms resemble the flu, doctors must rely on laboratory evidence to confirm the disease, using serologic testing or tissue staining to detect the presence of the virus. The virus causes extreme microvascular permeability, making your blood vessels leak. This leads to rapid systemic shock, leaving intensive supportive care as the only medical option. In the ICU, medical teams provide intubation and aggressive oxygen therapy, meticulously managing fluids to keep the patient alive while their immune system fights back. Since medicine cannot cure it, we must prevent ecological crossover using the CDC's Seal Up, Trap Up, Clean Up protocol. The first 2 pillars focus on physical barriers. You seal up entry holes to keep rodents outside, and you trap the ones that manage to enter your living space. The absolute golden rule of prevention is never to use a broom or a vacuum to clean up dry rodent trappings. This instantly creates the deadly aerosols that cause infection. Instead, the correct cleanup protocol involves thoroughly soaking the area with a bleach solution to deactivate the virus before wiping it away. Hantavirus is a masterpiece of viral evolution. However, understanding its biology removes the mystery and gives us the power to prevent it. Thanks for watching. Please like and subscribe. Computational Genome Biology.